continuamos con nuestros seminarios en el nuevo horario a las 9 de la mañana, todos los jueves, nada más como recordatorio. Y bueno, agradecemos a todos su presencia. En esta ocasión solamente tendremos un seminario, ya que una de las alumnas que le correspondía presentar hoy está enferma, nos envió su comprobante médico, entonces no presenta. Así que Sara tendrá un poco más de tiempo para las preguntas. Sobre todo para preguntas, observaciones o dudas que quieran ustedes hacer a su trabajo. Y bueno, me voy a permitir presentarla, Sara Frin, del 5º semestre, cuyos directores de tesis son el doctor Antonio Luna y el doctor Píndaro. Adelante, Sara, con tu presentación, por favor.
Okay, thank you very much. Hello, good morning, everyone. This is Sara Frin. Welcome to my presentation. I am going to tell about my thesis advance. Some problem. Sorry, and the title of my thesis is Effect of Probiotic and Quorum Quenching of Bacillus species on the Intestinal Microbiota, Survival and Immune Response of White Shrimp, Phineas venomii, challenged with Vibrio parahemolyticus, the causative agent of acute hepatopancreatic necrosis disease. The disease first emerged in China 2009, then Vietnam 2010. The disease reported from Mexico 2013. Here we see healthy shrimp and AHPND infected shrimp. From this figure we see healthy shrimp has full gut and stomach and large pigmented hepatopancreas. On the other hand, infected shrimp has empty stomach and mid-gut and pale hepatopancreas, and infected shrimp has another symptom, that is lethargy, slow movement. Shrimp immune systems has two defense mechanisms, innate immune system and physicochemical barriers. Innate immune system consists of hemocytes and humoral effectors. They have subcategories also. Bacterial quorum sensing is a process by which bacteria can communicate with each other through small signaling molecules called autoinducers and increase their population density. On the other hand, quorum quenching is the opposite process by which certain bacteria can produce some proteins or enzymes that can block autoinducers and thus inhibit quorum sensing. So this process can be used to control bacterial infection or biofilm formation. Previous studies showed that probiotic bacillus used to disease control and also growth promotion in shrimp. General objective of my study is to determine the effect of bacillus species in the expression of virulence factors of Vibrio parahemolyticus and its impact in the intestinal microbiota growth, survival and immune response of white shrimp cultured in the laboratory. Specific objectives are to characterize at a biochemical and genetic level the virulence factors of Vibrio parahemolyticus, to evaluate the effect of Vibrio parahemolyticus IPN GS16 added in water on the intestinal microbiota of white shrimp, to characterize at a genetic level the ability of adhesion and biofilm formation of different species of bacilli, to evaluate the effect of bacilli added in water and feed on the intestinal microbiota of white shrimp, to determine the protective effect of bacilli in white shrimp cultured in the laboratory and challenged with Vibrio parahemolyticus, and to evaluate the immune response of white shrimp that cultured in the laboratory treated with bacilli and challenged with Vibrio parahemolyticus IPN GS16. Hypothesis is the bacilli reduce the virulence of Vibrio parahemolyticus, change the intestinal microbiota, and increase growth, survival, and immune response of white shrimp. Justification: the disease causes serious economic losses in the northwest region of Mexico. Antibiotics are used in shrimps, but misuse or overuse of antibiotics can generate antibiotic resistance in Vibrio parahemolyticus. On the other hand, probiotics are environmentally friendly, so we should work with Bacillus species to observe their effects on white shrimp against the disease. General scheme of my work: bacterial culture conditions, biochemical and metagenomic analysis of Vibrio, determination of Vibrio virulence factors, antibiotic susceptibility test against Vibrio, antagonism of Bacillus species against Vibrio, determination of Bacillus species probiotic factors, cloning and sequencing of the acyl homoserine lactonase gene, disruption of Vibrio biofilm by Bacillus cell-free supernatant, bioassays with shrimp, metagenomic analysis of Bacillus, and statistical analysis. I had already tell about the biochemical characterization and metagenomic analysis challenged bioassay. Today I am show my recent work. This is my previous work and methodology according to biochemical characterization. For antibiotic susceptibility test, for antimicrobial sensitivity test, bacteria grown into TS broth and inoculated on the media. These 6 antibiotics placed on the media and incubated. When clear zone found around the antibiotic disc, it indicate positive result. Antagonism of Bacillus species against Vibrio parahemolyticus. Bacillus and Vibrio grown in Jobel marine broth, then Bacillus centrifuged and cell-free supernatants of Bacillus passed through 0.45 micrometer pore size filters. 10 microliter of Vibrio strain mixed with 10 milliliter of melted TSA, 50 microliter of Bacillus cell-free supernatants poured into the well and incubated. Positive result, clear zone around the well indicates positive result. Results. Total dead and survival shrimps from the treatment recorded from tank 1 survival shrimps were 12, tank 2 15 and tank 3 8. The number of dead shrimps from tank 1 was 13, tank 2 10 and tank 3 7, and the total number from each tank was 25. From microbial adhesion test, microbial adhesion to solvent test, found most of the strains showed moderate capacity of hydrophobicity. According to Lopez-Leon et al. 2016, Vibrio parahemolyticus showed moderate hydrophobic and hydrophilic capacity, and our results support their findings. From autoaggregation test, high autoaggregation values ranges from 94.83 to 98.21% were observed in Vibrio parahemolyticus strains. Co-aggregation ranges 0.97 to 55.28% were observed in different Vibrio parahemolyticus strains with Bacillus licheniformis. According to Ischemia Montes et al. 2015, co-aggregation of Bacillus with Vibrio sinaloensis was 66.6%. And most of the strains showed medium biofilm capacity. IPNGS 16, 21, 24, 28, and 30 showed high capacity of biofilm. These are the strains. Mahdi et al. 2010 reported that probiotic Bacillus species have biofilm capacity, and our result showed that pathogenic Vibrio Vibrio isolates biofilm formation capacity have a biofilm formation capacity also. Most of the strains showed medium growth under 0.5 to 5% concentration of sodium chloride. Strains 4, 29, 30, 31, 32 can grow in all salinity, whereas Ischemia Montes et al. 2015 showed that Bacillus can grow well from 0.5 to 9% of sodium chloride. And most of the strains can grow under pH 6 to 9. Well growth was found in neutral pH. Most of the bacteria prefer a neutral pH, according to Tuomola et al. 2001, and Bacillus BCR423 also grow better at pH 6 to 7 from Ischemia Montes et al. 2015. Most of the strains showed exponential growth phase in between 6 to 12 hours. Lopez-Leon et al. 2016 showed that the log phase of Vibrio isolates was found between 12 and 96 hours, but hours found between 6 and 12 hours that different from their findings. The range of CFU per milliliter was from 1.1 into 10 to the power 6 to 64.8 into 10 to the power 6. Antimicrobial sensitivity test from these results, all 33 strains were sensitive against the antibiotics sulfamethoxazole, enrofloxacin, oxytetracycline, and fluorphenicol, except strain 14. 21 strains were resistant against streptomycin. Antagonism of Bacillus against Vibrio parahemolyticus. Bacillus licheniformis showed high antagonism ability against strain 4, 9, 15, and 27. Next generation sequencing were performed for intestines of white shrimp. The total reads obtained from the NGS were 685, 972 reads. High quality reads were 559, 274. Total O2s were 406. Taxonomic assignments at 97% identity with the Silva database. Reads obtained from Shaman and O2US from microbiome analyst platforms. Relative abundance of phylum from microbiome analyst. Here A is control or non-infected shrimp before infection, and B dying shrimps with challenge, and C surviving shrimps with challenge. From this, we see microbial community structure showed that Proteobacteria was the most abundant genus, followed by Bacteroidetes, and the abundance of Proteobacteria is highest in dying shrimps, but the abundance of Bacteroidetes is highest in survival shrimps compared to control and dying shrimps. And this result correlates to Gainsa et al. 2017 and Cow et al. 2019. Vibrionaceae, Flavobacteriaceae, Rotobacteriaceae, Pseudo-Alteromonaceae are the most abundant family. At genus level, we see Vibrio, others, FIFO, Nautella, Pseudo-Alteromonas are the most abundant genus, and the abundance of Vibrio is highest in dying shrimps compared to control and survival shrimps. Cheng et al. 2016 established high relative abundance of Vibrio, Rugeria, Pseudo-Alteromonas genera that correlates with our findings. This table shows the percentage relative abundance of microbiome analyst. Here we see the abundance of Proteobacteria was more than 80%, and that is similar to Vargas et al. 2017. All this family, the abundance is decreased in dying shrimps except Vibrionaceae, and the genus showed the same result. Vibrionaceae, the abundance is in dying shrimp is 88, and the abundance decrease in survival shrimp to 13.4 on, and control condition is 53.23%. Alpha indices. Alpha indices analyzed the species diversity, that refers the number of species in a community and the abundance of each species. Shannon and Simpson analyzed the number of species and abundance, and Chawan and AC analyzed only the number of species. And all the four index showed that the number is highest in survival shrimps and lowest in dying shrimps. Rare fiction curves showing species richness and sequence sample size. From this curve, richness found in survival shrimps and poor richness found in dying shrimps. Beta diversity measures the change in diversity of species from one environment to another. It calculates the number of species that are different in two environments. The components explain the 90.6% of variation. The samples are grouped into three groups, but each group showed wider intradispersion. The principal coordinate analysis showed that bacterial communities in the shrimp gut in control and treatments are significantly different. Some problem. Okay, sorry. Bacterial community of shrimp intestine had a high content of genes related to metabolism, genetic information, organismal systems, etc. Metabolism are highly represented, that is similar to Wang et al. 2015. Some problem. Okay. These are the metabolism functional categories like amino acid, carbohydrate, lipid metabolism, etc. According to Wang et al. 2015, the presence of these metabolism functional categories indicate that the metabolic potential of bacteria is highly diverse, and they are well adapted for the degradation of carbohydrates and amino acid. These are the top metabolic functions. Here we see quorum sensing related metabolism showed highest percentage in dying shrimps, that is 17% compared to control, 14%, and survival 11%. Functional interaction networks. This figure shows the functional interaction networks between genera of different phylum. The different color of nodes, indicate the genera belongs to different phylum. Here we see red nodes and olive green nodes. That red nodes indicate Proteobacteria and olive green nodes indicate Bacteroidetes, etc. Blue lines indicate positive interactions and red line indicate negative interactions. The core generated by 9 genera. For dying shrimps, we see Vibrio dominated the network core. Here and there are some large nodes but with fewer interactions. In the case of survival shrimps, there are a highest number of large, medium, and small nodes, and we see highest number of interactions. The network core was formed by 13 genera. Preliminary conclusions of my studies are biochemical characterization showed a high pathogenic potential, especially strains 16, 21, 24, 28, and 30 with a high biofilm formation capacity. The phyla Proteobacteria and Bacteroidetes predominates in all shrimp intestine samples. At genus level, Vibrio was higher in dying shrimps and lower in survival and control shrimps. Ruggieri and Pseudo-Alteromonas predominates in survival shrimps, and they are probiotics in animals. Less bacterial diversity was found in dying shrimps. 6 functional categories are found in all samples. In dying shrimps, quorum sensing related metabolism showed the highest percentage. Functional interaction networks, negative and positive, are higher in survival shrimps that I showed and lower in dying shrimps. In survival shrimps, the network analysis showed more core nodes 13, then control condition 9, and dying shrimps 1. In dying shrimps, Vibrio dominates the network core. Limitations of my work. The lab activities were interrupted due to COVID-19. If Bacillus does not work against Vibrio, we will need to find other isolates that will take a long time. From August to October, I have worked with my article, and currently it sent to the Journal of Aquatic Research in October 11. This is the chronogram of my activities. Thank you for your kind attention.
Thank you, Sara.
Thank you.
Ahora es tiempo de preguntas. Alumnos,
Okay.
por favor. ¿Tienen preguntas para Sara? Las pueden hacer en español. Entiende bastante bien Sara, y si no, le ayudo por ahí con la traducción. Así que anímense, muchachos.
Yo, doctora.
Adelante, Karime.
Hi, good morning, Sarah.
Hello, how are you?
Thanks, how are you?
Fine.
That's good. I wanted to ask you if you could tell me a little bit more about quorum sensum and quorum quantum, please.
Okay. Actually, quorum sensing is a regulatory process by which bacteria within bacterial cell, one cell to another, they can communicate through these small signaling molecules that called autoinducers, and by this they can communicate, and they can increase their population, they can increase their number, and it show virulence against it show virulence, and it cause bacterial infection, and quorum quenching inhibit the process of quorum sensing, because some bacteria can produce some proteins or enzymes that can block these inducers, because enzyme has very specific for these autoinducers, and it can block. So for this, the bacteria cannot communicate with each other, because the autoinducers blocked by the enzymes. So the process is inhibit, and that's why I tell this is effective for disease control and biofilm formation.
And proteins or these enzymes are produced by the same organism or by other organisms?
By the other organism that is beneficial for shrimp or.
Oh, okay. Thank you, Sarah.
Thank you. Okay.
Perdón. Paulina Montserrat, adelante, por favor.
Hi, Sarah. In your presentation in number 16, I have a doubt because you only showed tank ABC and showed the survival and that one. What's the difference between each tank, or what were the treatments between them?
Okay, that there are three treatments. First time zero, that I tell that I had already tell or showed in the previous semesters. That's why I don't show here. There are three treatments, and we counted time zero. for fast treatment, and that shrimps are not infected with Vibrio parahemolyticus. This treatment for those three tanks. Every treatment, the shrimps are collected from these three tanks, and after infection, after 24 hours, we again collected shrimp intestines, and that time we collected dying shrimps. That shrimps were vulnerable with the infection, that shrimps. And finally, after 74 hours of infection, we collected the shrimps that can survive in spite of the infection. And we monitoring how many shrimps dead, and at last is how many shrimps are survived. These are the numbers of that shrimps. Maybe I can explain.
Okay, thanks.
Thank you.
Cari, ¿me tienes otra pregunta?
No, doctora, ya bajé la mano. Gracias.
Gracias. ¿Alguien más tiene preguntas para Sara? ¿Es turno también para profesores? Ignacio Maldonado, adelante, por favor.
Hola, en realidad yo quisiera tomar la palabra. Mi máquina no se quiso conectar al Teams, entonces aquí estoy.
Adelante.
Hi, Sarah. Nice talk.
Hi.
I have a suggestion.
Thank you.
Nice to meet you. Yes. I have a suggestion and a question. My suggestion is that maybe you can give us, can you understand? Maybe can you make it difficult for you to understand?
Yes, doctor, yes.
Okay. So my suggestion is to ask you please to give us maybe for the next presentation a little bit more information in the titles of your slides. You know, you have a lot of information, you have a lot of data, and sometimes it's hard to follow because, and this can be fixed if you just can give us a little bit more information on the title of your slides. Okay?
Okay, doctor, thank you.
And my question is that, so you checked for like the aggregation capacity of your Bacillus strains, right? Am I understanding correctly?
Antagonism, doctor?
Yes, aggregation. Aggregation and film.
Yes. Aggregation of Vibrio. Yes.
Aggregation of Vibrio. Okay. So for the Bacillus strains, you only check antagonism?
Sorry, doctor, please ask again.
Yes, for the Bacillus strains, did you check for antagonism? You check aggregation and film formation for Vibrio, right?
Yes.
Okay. Yes. And antagonism, did you check it for Bacillus, or did I understand wrongly?
Okay.
So you have 30 something, 33 Bacillus strains or Vibrio strains?
Vibrio strains.
Then I was confused. Okay.
These all the strains of Vibrio on 233, and Bacillus licheniformis strain. And we test antagonism of Bacillus against these strains of Vibrio, all of these strains of Vibrio. So this is antagonism of Bacillus. That if Bacillus can, sorry, not this. bacillus can inhibit Vibrio or not.
Okay.
We check this or test this.
Okay, I got this confused. Okay, thank you.
Oh, okay.
My other question, just to keep going, is what is the most important result? What do you that you expected to have from your metagenomic analysis?
From metagenomic analysis, all the results, the summary of my results are important for me, and all the results show that Vibrio change the or effect on the intestinal microbiota. Because in survival shrimp, it impact on the metabolism, and in dying shrimps also diversity. In case of dying shrimps, diversity is lowest, and in the survival shrimps, it higher and more. There are more diverse bacteria or different bacteria, many different bacteria in survival shrimps, and I think this because Vibrio change the environment or change the bacterial environment.
Yes.
After infection.
Great. Okay. Good. And a very technical question: how many replicates did you do for the metagenomic analysis, or did you make a pool, or did you make did you sequence replicates?
Yes. We made pool, and for every treatments. First we collected for normal shrimp that not infected with Vibrio parahemolyticus, and we collected 3 shrimps from 3 tanks, total 9 shrimps, and for tank 1 we made a pool, tank 2 we made a pool, total 3 pools, and every treatments we follow the same procedure.
Okay, all right. Thank you, Sarah.
Thank you, Doctor.
Thank you, Melina. Si no tenemos más preguntas, ¿algún otro profesor, alumno que se quiera animar? De no ser así, vamos a dar por concluidos los seminarios del día de hoy. Y agradecemos la participación de Sara. Y pues nos vemos el próximo jueves. Recuerden, tenemos cambio, nuevo horario. Y les recuerdo también las evaluaciones. ¿Sí? Queda la evaluación para Sara. Y pues nos vemos el próximo jueves. Thank you very much, Sara.
Thank you, Doctor. Goodbye.